Jens Wildfang is a graduate.
That's it.
Wildfang. Jens Wildfang is a graduate from the Faculty of Medicine of Hanover, where he obtained a PhD in psychiatry. He is the director of the Department of Psychiatry and Psychotherapy at the University Medical Center in Göttingen. He's a neurologist and psychiatrist with wide experience in the field of neurodegenerative diseases. His activity includes the clinical characterization of patients with dementia and research on the biomarker discovery and validation. In particular, his main expertise is on the field of cerebrospinal fluid and blood biomarkers for early diagnosis of neurodegenerative diseases, with special interest on Alzheimer's disease and Parkinson's disease. So, I'm very interested in that. So.
Yeah, thank you for the kind invitation. So I'll start right away. So the topic of my talk is predictive molecular diagnosis as a requisite to set up novel clinical models for preventive treatment. And why is that? So current needs are treatment of Alzheimer's disease starts much too late, and most importantly, there's strong evidence that we miss critical time windows. We definitely need novel preventive treatments. What we have available nowadays is purely symptomatic. We need to identify novel subphenotypes of multigenetic sporadic AD. They are genetic cases, but they make up only 1% of also cases. So the predominant cases are multigenetic sporadic cases, which may then respond differentially to treatment interventions. And in this area, biomarkers are very promising. And last but not least, we need to identify novel treatment targets. And I will give an example in my talk that research primarily dedicated to diagnostics may come up with novel therapeutic targets as well. Now, this is the amyloid cascade hypothesis of Alzheimer's dementia. Our group does not claim that this is the etiology. For multigenetics, this is still open for debate, but at least those biomarkers are highly correlated surrogate markers, and thus they can be used for diagnostic terms. So here you have the amyloid precursor protein with a large extracellular domain, which then is cleaved by beta and gamma secretase to give rise endoproteolytically to a beta peptides. Two species are depicted here. Actually, this is much more complex, as I will deline later. Beta peptides are prone to form soluble oligomers via amyloidogenic intermediates. We will tackle this by a new assay, which I will present at the end of my talk. So this is molecular neuroimaging. And then soluble oligomers precipitate as insoluble amyloid fibrils, which finally form the beta amyloid plaques described by Alzheimer. And correlated to this precipitation is a selective decrease of 1-42 in cerebrospinal fluid, because 1-42 makes up the majority of those fibrils and plaques. So that's an important take-home message. We have a selective decrease of 1-40 because 1-40 is more or less unchanged. So the ratio 1-40 is declining in CSF. Then in the vicinity of the plaques, we have a strong neuroinflammation, which is a promising novel therapeutic target. And within the neurons, we have the formation of neurofibrillary tangles, which have also been described by Alzheimer by staining techniques. Of course, he didn't know the molecular constituents. And with emerging synaptic and later neuronal degeneration, we have a release of the molecular constituents of neurofibrillary tangles. This is tau protein, most importantly, hyperphosphorylated tau protein species. So second take-home message, in CSF, tau goes up, more specifically, phosphotau. Total tau is a more unspecific measure of neuronal destruction. Then those patients who are on their way to dementia, unfortunately, they have to pass a preclinical state of mild cognitive impairment, where cognitive deficits are evident, but they are not so strong that they do not deteriorate everyday life performance. Now, this is a famous landmark paper this is supposed to be Hansen in Lancet Neurology from the group of Kai Blendo in Sweden, who they conducted a multi-center study where patients at several clinical sites presented in the prodromal state of MCI. Then the CSF dementia biomarkers were estimated at baseline 1 to 40 total tau phospho-tau, and those with a normal signature had a very less risk to progress to AD, whereas those presenting already preclinically at the prodromal state of mild cognitive impairment with this molecular signature of AD had a high risk to develop AD within the next years, and this corresponds to positive and negative predictive values of roughly 90%. So negative and positive predictive values are completely meaningless without data on prevalence, but the prevalence in the MCI cohort of later Alzheimer's is roughly 20 to 25%. So in view of a prevalence of the hidden prodromal ED of 20-25%, this is highly meaningful in clinical terms. Now, these cerebrospinal driven neurochemical dementia diagnostics of ED has meanwhile been validated by several autopsy-controlled studies. Meta-analysis revealed specificity and sensitivity of roughly 85% in more than 20,000 patients worldwide in several independent multi-center studies. And due to this body of evidence, this diagnostic has entered national and international diagnostic guidelines. And meanwhile, research is dedicated to improve the performance regarding coefficients of variation, bias, etc. And there are large EU research initiatives dedicated to this, like Biomark ADP. Now, it's not only useful for the predictive diagnostics, but as you will see here, it can, to some extent, predict the acuity of disease progression. Look at this. So those are patients with a very rapid progression from MCI to AD, those with intermediate speed of progression, and those are slow converters. So there's no correlation of the decrease of 1 to 42. It's decreased, but it's not correlated to the speed of deterioration. Those are the controls. So this is completely different for phospho-tau and total tau. Those presenting at baseline with high tau levels are much more at risk for a speedy deterioration. Now, this is another very important landmark paper of Bateman and colleagues published in New England Journal of Medicine recently, and they investigated those biomarkers, including imaging biomarkers like amyloid PET or glucose PET and structural MRI in a large set of patients with genetic autosomal dominant PRISM1 mutations. And it became very clear that the first changes of biomarkers, look at 1 to 42, it appears 25 years before clinical manifestation of the disease. First, there's a paradoxical increase. It's not paradoxical, actually, but there's an increase. Then 20 years for clinical manifestation, you hit the cutoff line of the population, and when you can diagnose the disease, it's almost at the level of decrease and doesn't change anymore. And if you look at a better deposition in the brain now, which now can be measured by F18 amyloid PET, you already reach the saturation when you can diagnose in clinical terms the disease. Now, so this is CSF, but we would like to have blood assays, and I will give 2 examples from our own research. There's another paper and very promising data from the group of Simon Lovestone from the UK, but due to lack of time, I cannot detail this. So this is a method which some of you might know, at least those working in the field of neurodegenerative diseases. This was developed when I was working at the Max Planck Institute in Göttingen for several years before I started my clinical career. And it relies on the fact that beta peptides change their conformation in the presence of urea due to modulation of intramolecular H bondage. So it's not, in this case, the separation principle is not sieving of the SDS-PAGE gel, but induced conformation changes, which are highly correlated to the primary amino acid sequence and post-translational modifications of beta peptides. And this is really specific for beta peptides. Other groups of peptides do not show this peculiar finding. So first, there's a separation with isoelectric focusing, with immobilized pH gradients, and then we have this urea-based second dimension, followed by Western immunoblot with the attomolar detection sensitivity, so this corresponds to several picograms per spot. And we were very surprised that in blood plasma of the patients, the pattern is much more complex than the CSF of the same patients. So this is a 1 to X species, let's say 1 to 37, 1 to 38, 1 to 39, 1 to 40, 1 to 42. So as you can see, you can separate peptides only differing in a single amino acid, which is completely impossible by a 10% T and 5% C separation gel. But to our surprise, there was a reproducible, another lane of beta peptides corresponding to the truncated, aminoterminal truncated species 2 to X, so 2 to 37, 2 to 38, 2 to 40, 2 to 42, missing the aspartate, so we have a shift in the isoelectric point. But at exactly this position, we have a co-migration of the elongated -3 to 40. And I will come back to this because we have quantitative mass spec LC tandem MS data from colleagues from Japan actually pointing to the fact that this is more or less solely -3 to 40. So now if you look at this novel ratio, 1 to 42 divided by the elongated -3 to 40, we were able to correctly classify 80% of patients with early ED, and most importantly, of those preclinical amnestic MCI patients, and those patients were cross-validated by their CSF biomarkers. So we know that unfortunately those patients, due to their CSF biomarker signature, are at high risk to develop AD, and so this is also capable of identifying via blood-based diagnostic those preclinical patients. This is a receiver-operate curve characteristics for the standard ratio, the gold standard ratio, and the novel ratio. Now this is a brand new data. So this we patented in 2010, and meanwhile there's this very, I think, strong paper from Japan of a cohort of roughly 60 patients. Every single patient had been cross-validated by F18 amyloid PET. So in clinical terms, this is a very sound diagnostics. And as you can see, you can nearly distinguish the receiver-operate curve characteristics of the F18 amyloid load, but a single scan costs 2,000 euro, and the assay may cost in the end 30 euro. It's almost impossible to distinguish receiver-operate curve characteristics of F18 amyloid PET and the novel ratio. And the novel ratio is clearly superior, like in our data, as compared to 1 to 40 in the gold standard ratio 1 to 42 divided by 1 to 40. This is the mass spec data. So this is immunoprecipitation followed by LC tandem MS. You see this is a prototypic pattern in control, and this is a prototypic pattern in an AD patient or preclinical patient with a relative increase of the elongated -3 to 40. Now, here you can see that in contrast to 1 to 40 and the gold standard ratio, the novel ratio is clearly correlating with the F18 amyloid plaque load. Now, in a completely different line of experiments, in experimental models, and human stem cells, neural stem cells. I will not bring those data due to lack of time. So these are data in S555 cells where we were very surprised if we inhibit the beta-secretase base, which is responsible to give rise for this 1-to-X species. As expected, we have a clear decrease of those species, let's say 1 to 40, there's almost a shadow left. But to our surprise, other species were not decreased, but even paradoxically increased. And so this theoretically could be 2 to 40, but now we validated this with mass spec, and actually this is -3 to 40. So there's evidence that those novel species are generated by a novel, so far uncharacterized beta-secretase, which is a very promising novel target. And this may be the reason why the novel ratio depicts a completely different information. It depicts information of the endoproteolytic cleavage balance of 2 different enzyme systems, whereas the gold standard ratio 1 to 42 divided by 1 to 40 is within base. So that's a completely different set of information, and this may explain why it's superior. So I can jump this. So the clinical significance of those aminothermally modified peptides is that they're highly abundant in human brain. We demonstrated this in JBC before. And that, most importantly, transgenic animal models and human AD brains differ dramatically in the composition of those species. So they are, in relative terms, 10-fold more truncated or modified species in human brain. And they may form a seed for seeded aggregation, so a kind of multiplication procedure or principle as known for prion diseases. Now closing this up, we published in Science 2009 that there is not a single gamma secretase, but there are distinct gamma secretases which give rise to different beta peptide species. So could it be that we have also a distinct beta secretase activity as depicted here? I think our data and data from Eric Portelius and others give clear evidence that this is the case, and those may then be novel therapeutic targets as well as diagnostic targets. Now, the last two folds, the last 2 slides, we recently developed a completely new immunosensor relying on Fourier transformed near-infrared spectroscopy as a novel biomarker for prion misfolding diseases. It works the following. On top of a germanium crystal, as part of a flow-through cell, we have antibodies immobilized which catch the antigen, in this case a beta peptide, but it also works for alpha-synuclein, for Parkinson's, for example. And then using Fourier transformed spectroscopy in the near-infrared spectrum, you can have a quantitative readout of the secondary structures. So what percentage is random coil, what percentage is alpha helical, and most importantly, what percentage is beta sheet folded. And our data clearly point to the fact that already at the level of the monomer, patients with AD have a clearly increased ratio of this wrist folding state, which is a prerequisite for following oligomerization in CSF, but most surprisingly also in blood. So these are the CSF data which correspond to a classification accuracy of roughly 90%. Now to sum this up, the molecular pathophysiology of Alzheimer's disease is launched more than 10 years before cognitive brain deficits of Alzheimer's dementia become overt as amnestic MCI. Beta-amyloid-mediated neuropathology with neurotoxic soluble oligomer species precedes tau-mediated neural damage, and moreover, there's a strong evidence for a prion-like disease propagation. A gain of endoproteolytic function in rare autosomal dominant genetic AD is well documented. However, in those multigenetic, 99% of the cases, scientific evidence points much more to a loss of catabolic clearance function. In case of those multigenetic cases, the final molecular cause has not been validated yet. but are better and tau are at least highly correlated surrogate markers, and we can make use in diagnostics of this fact. Biomarkers can identify preclinical AD, which allows to validate promising preventive treatment strategies in those clinical models. And finally, aminoterminally modified abetapeptide species are likely to be generated by non-base secretase activity, and they may offer new diagnostic and therapeutic molecular targets. Thank you.
So today we speak about a comprehensive pipeline for biomarker discovery using circulating microRNA microarray data. This is the outline of my presentation. I will briefly introduce the high-dimensional omics data. Then I will speak about our methodological proposal for data analysis. Then I will show you a case study, the 4-HPR INT study. And then there will be space for discussion and future perspective. Omics data are various and range from genomics, macrobiomics, proteomics, and so on. In particular, in the field of macrobiomics, mRNA-omics, we developed a project that is called IDERA. Sorry. Thank you. Which is acronyms of Early Detection and Risk Assessment. And it's sponsored by the Italian Association for Cancer Research and is related to the tumor microenvironment-related changes as new tools for early detection and assessment of high-risk disease. So, you know that microRNA are highly conserved single-stranded small RNA molecules with key role and also in cancer development and progression. To date, more than 2,600 human microRNA have been identified. And moreover, several independent studies have reported the feasibility of using plasma microRNA as molecular tools for detection, prognosis, and treatment decision-making of various cancers. However, some biological and technical challenges could limit the development of microRNA as biomarkers. So, the aim of our methodological study was to develop a multi-marker hemolysis classifier using plasma microRNA data. In particular, from a methodological point of view, we want to propose a strategy, and we applied it to hemolysis because it is a particular problem in sample processing, very common. And so it's important to also to validate this strategy. So we choose hemolysis also for the availability in literature of the results. So we know before which microRNA should come out. So it's a check. This is the workflow of the strategy. In particular, we start with sample processing from study design to microarray hybridization. Then we have data pre-processing with microRNA filtering and also the consideration of raw and ratio normalized data. And then we have the supervised data analysis with class comparison and class prediction analysis. As regards study design, we consider a subset of patient samples from a randomized breast cancer prevention trial, from the control arm in particular, with 1,476 patients. We extracted RNA subjected to Agilent microRNA hybridization array, and we choose the Agilent system as a microarray platform, getting 1,205 human microarray. Hemolysis was evaluated with the hemolysis score, which is a score that was developed in the context of the IDERA project. And we compared hemolyzed versus not hemolyzed plasma samples according to this score. Then we 1-to-2 matched the samples using the nearest neighbor method, using as matching variables disease status, age at drawing, and drawing year. So we got the training and validation set. Regarding data preprocessing, each mRNA was targeted by 1 to 4 different probes, and each probe spotted 10 to 40 times on the array. The total signal for each macroarray was obtained by summing the probe signals derived from Agilent features extraction software. We then log transformed the data, filtered them at 90%, and normalized them with a ratio-based approach that was developed inside also this project, the DERA project. Then we considered both raw and ratio normalized data. Regarding class comparison, two sample location tests are commonly used as univariable feature ranking methods. However, in this context, we have very non-normal distribution, very particular ones. So skew, tail, multimodal, even after data preprocessing. And this gets worse with small sample sizes. So location tests seem not adequate to identify differentially expressed features. More general distributional discrepancies might hide differential biological processes. So we didn't consider only the t-test, which looks only to location differences, but we considered a non-parametric Anderson-Darling test, which is an omnibus test that considers the whole feature distribution, getting more importance to the observation in details, which is very common. This is an example of the shape of the distribution you can get from data like this, like omics data. So it's very important to also keep features that have like something like that, a shape, a particular shape in the tail of the distribution. Then we used the Benjamini-Hochberg method in order to adjust the p-values from both the two tests. And then we combined the results of the T and the Anderson-Darling test, considering significantly differentially expressed those features with FDR adjusted p-value below the 5% level for at least one of the two tests. Regarding class prediction, we drew 1,000 bootstrap samples from the original dataset, and we ranked the feature according to the joint selection by 3 different classification machine learning algorithms. In particular, we considered the PEM, prediction analysis for macroarray, which is a centroid-based method, the random forest method, which is a tree-based method, and the elastic SCAD SVM. Why? Because these are the representative of 3 different methodological categories. So, for example, PEM is very fast and simple, but it cannot deal with complex patterns. A random forest can deal with complex patterns and it's also fast, but it cannot deal with correlated features. And so there is the choice of the elastic SCAD SVM. So we graphically summarize the results with an X-shaped plot, which shows both bootstrap occurrences and co-occurrences. In step 2, we developed several classifiers, in particular several different linear SVM models in a Lee-1 autocross validation loop, optimizing at the same time the SVM parameters and the number of features. So we got the Lee-1 autocross validation sensitivity, specificity, and obviously the Uden index, which is the sum of the sensitivity and specificity minus 1. Then we chose the final model according to the highest Uden index and the smallest number of features to be more parsimonious as possible for practical purposes. And then we graphically summarize the results with a rock space plot. We call this because it's a sort of generalization of the rock curve. And this shows the classification performance for all SVM models. So, getting to the results, we finally, after the choice of samples and the evaluation of the homology score, we selected 78 samples, 26 hemolyzed, and 52 not hemolyzed. Then we also split them into a training set and a validation set, and considered 88 macroRNAs and the corresponding 3,828 ratios. So, regarding class comparison, we plotted the classical volcano plot with the t-test and the corresponding effect measure, the fold change. But we also considered another type of plot, a sort of concordance plot between the t-test and the Anderson-Darling test. And if you look there, in this case, for raw data, there were 3 macroRNA in common between the 2 tests, and one discovered only by the Anderson-Darling test. And for ratio data, the difference is even more. So 170 for both 2 tests, 28 for the t-test, and 26 for the Anderson-Darling test. Oh, I'm sorry. Class prediction, so it's briefly the ranking of the features. So I highlighted in the red the top 5 features. And we also have the bootstrap co-occurrences. Then this is the S-shaped plot. The larger the node, the more important is the feature. The thicker the edge, the more important is the bootstrap co-occurrence. This is the rock space plot. Briefly highlighted in red the most important models which corresponds to the highest performance. Then this is a summary of what we got, so different models from the Liguanato cross-validation with their Uden index and the number of Myrna included into the models. And then we evaluated them in a validation set. So to conclude, we developed a general analysis strategy to deal with some issues arising in the supervised analysis of plasma macroRNA from hybridization array data. In data preprocessing, we use the ratio method as a normalization method because we didn't want to choose any reference Myrna. So there is automatic handling of a specific normalizer for each differentially expressed macroRNA. In class comparison, we used the Anderson-Darling test as a new test, possible test for class comparison. And it's important to pay attention in using in class comparison this type of univariable statistical analysis for ranking purposes. In class prediction, we obtained robust results from the bootstrap selection and the classified cross-validation. The S-shaped plot was a very beautiful means to give an insight on the interconnection among the selected features. And we also pay attention to the choice of the most parsimonious models. We identified 5 Myrna, I said before, the 4 reported in literature, and sorry, and also the Myrna 22, a new one. Okay, briefly, we have some study limitations, small sample sizes, lack of ability of an external data set, a testing set, and also the fact that the SWM uses surrogated class probabilities, which are not the classical ones. We applied also it to other types of data with proper adjustment. And so finally, to conclude, future perspectives are the search for an alternative normalization method, the use of the Anderson-Darling test for the construction of a volcano plot with a corresponding effect measure, and also the fitting of other types of models for classified development. This is the acknowledgement to our group, the people who work with us. Thank you so much for the attention.
Thank you for the introduction. My name is Jin Zhan. I'm from Royal Military College. I work as a postdoc fellow. And this talk is about insights to use the microRNAs from mammalian hair follicles to be used as a traumatic brain injury biomarkers. So traumatic brain injury, TBI, is described as any brain injury caused by physical impact to the cranium. So it is a very common disorder. Actually, it happens every day, and you can see that happening in competitive sports like a hockey game. The concussion leads to the injury. And of course, I am from a military institute, and the focus is somewhat, focused on the army type of stuff, and they have the combat duties would lead to the use of explosives as weapons, so that the explosion would cause a shockwave to the head of soldiers. So that is a cause of a TBI in those type of situations. So it is very important to have some sort of system to serve as a study system as well as a diagnostic system. So, of course, the brain is very naturally used as a system to study this disorder because this is the site of the damage, but it is very hard to get access to. So people start to use blood as a biomarker system because it's relatively easier than brain to get. Still, it has to you have to have like the professional skills to take the blood. So our attention turns to the scalp hair follicle because for one reason is relatively easy to get because you just pluck the hair. Even I can do it. And also because the hair follicle actually goes through the development of the hair follicle goes through multiple cycles and multiple steps. It has a rich, it has the potential of housing rich information. So we decided to look into the potential of it to be a potential system to study TBI or serve as a biomarker system. In fact, we published our pilot study last year. So in this study, we used a microarray system platform to screen the gene expression profiles from the rat model whisker follicle to see if there's any shared gene expression patterns with brain or blood. And in fact, we found some shared results, and also because hair follicle is going through a unique process of, it has its own biochemical processes, so it has also its own unique molecular signatures. So for next, there are two objectives for this talk actually. One is next step naturally is to see how sensitive the hair follicle is to different intensity of the blast. And another one is to see if there's any potential new biomarkers other than messenger RNA. We tend our attention to microRNA. So basically what we did at Suffield base is that we developed an advanced blast stimulator. So basically what this thing does is it generates a single shockwave intensity to the animal model. Basically, we stabilize the rat into the shocking chamber of this tube. We shock them with different intensity shock waves. So we tested 15, 20, 25, 30 psi intensities, and after the exposure, we collect the whisker samples from the rats. So on the in the lab we do microarray and of course the other molecular tests. So for this purpose, we actually follow through this flow chart. We started using a Agilent microarray platform for these follicles collected from those different intensity exposures. And then we use a subnetwork enrichment analysis to construct the messenger RNA and mRNA interaction network. So based on the messenger RNA transcript levels from the microarray data, this test proposed the potential microRNA targets that could be responsible for those changes. And then we developed a qPCR-based approach to actually amplify those potential microRNAs to see if there's any real changes in the hair follicles. Of course, we have additional tests, but that's not this talk. So from the microarray results, actually, you can see from these volcano plots, the green dots in figures, they are the differentially expressed genes actually with a fold change greater than 1.5. You can see obviously as the increase of the blast intensity, the distribution of the quantity of the DE genes are different, obviously. And also the shared, the overlapping, the Venn diagram at the bottom shows that actually the overlapping DEGs are very scarce. So basically it says, what this thing tells us is that on the gene level, the hair follicle is able to represent, is able to respond specifically to individual intensities of the shock waves. Then we tended to a gene set level. We enriched the biological processes terms of the gene ontology terms, just to see the distribution of the enrichment. We use a page test for this test. As you can see from the heat map, the downregulation and upregulation of the GO term, obviously you can see it's different in each intensity shock waves. So at this level, we can also say that the hair follicle is able to enrich different biological processes, potentially, according to the blast intensities. So next is to see if there's any potential of microRNAs. Actually, the microRNA has been used as biomarkers because it has its own specific tissue-specific or developmental stage-specific expression profiles. And also, when it's circulating, it is usually within exosomes, so it is relatively stable to measure. And of course, they are very conserved. You can see this MIR26A, the sequence is the same across different species. So it is very easy to transfer the knowledge from the animal model to human subjects. In fact, as you can see here, of course, from this from the course of this conference, we saw a lot of studies exploring microRNA as biomarkers. So our strategy is to, as I said before, is to base on the microarray data on transcript on messenger RNA on the gene transcript to propose potential microRNAs, then to measure them to basically validate. So this one example is MIR183. This was proposed across all intensity shock waves. And you can see the central node is that microRNA, and you can see the blue colored shapes are the downregulated transcript levels, and red is the increase. So basically, the subnetwork says this microRNA could be responsible for these changes across all the intensity exposures. And then we measured this level, this microRNA in the hair follicle. You can see it's beautifully decreasing in all of them, and with a huge decrease in 20 psi. This microRNA has been found, according to other publications, has been found responsive to neurological disorders. So this one example is a shared microRNA candidate under the stress. And then this next one is only proposed in 15 psi exposure. And as you can see, the qPCR amplification suggests it was a decrease. And this microRNA has been proposed as a microRNA that's responsive to ER stress under central nervous system. The next example is at under 20 psi, MIR34A. This microRNA is very interesting because, first of all, it is specific to this intensity, and secondly, of course, it was downregulated in the hair follicle. And then, according to the literature, it was found once that in rat brain, it was responsive to TBI disorders. So basically what this thing tells us is that this microRNA, we can use hair follicle to detect the microRNA that's responsive to TBI. Sorry. So basically, we can use hair to get a similar microRNA pattern as a brain tissue. So basically, to summarize, what we found is that the hair follicle is, first of all, it's very easy to get. Secondly, it's sensitive to specific blast intensities. And then we explored the microRNA as a potential biomarker system because it's highly conserved, we can transfer the knowledge from the rat to human subject. And then we have shared microRNA. The example is miR-183, and to use the shared results, we can actually monitor the universal responses of TBI. And then we have intensity-specific microRNAs. And use this, we can actually see or monitor the damage progression of the TBI. So basically, yeah, that is our findings. And then these are the lab members. Thanks very much.
Hello, everybody. My name is Surasawadi Asawarat. I'm from Mahidol University, Thailand, and it is my pleasure today to talk about my research work entitled Efficacy of the TSH receptor mRNA in the follow-up of differentiated thyroid cancer, abbreviated DTC. I hope you find this topic interesting and informative. What I'm talking today are outlined here. First is the detailed information about the thyroglobulin as a tumor marker of DTC. Second is the review of the thyroid-specific mRNA as a surrogate or adjunctive marker. And third is my research on the efficacy of TSH receptor mRNA in the follow-up of DTC. And finally, my further study on the biomarker for DTC. Thyroglobulin is the glycoprotein dimer that help synthesis of the thyroid hormone. If the body needs the thyroid hormone, the thyroglobulin will be digested in the lysosome, and then the thyroid hormone are released to the bloodstream. The clinical significance of the thyroglobulin is that they are widely accepted as a tumor marker for differentiated thyroid cancer, particularly papillary thyroid carcinoma and follicular thyroid carcinoma. After thyroidectomy and radioactive iodine ablation, thyroglobulin levels can be serially monitored, and if there is an elevation of the levels, it indicates the residual thyroid tissue or recurrence or even metastatic disease. However, there are several limitations for the serum thyroglobulin. First, if the serum thyroglobulin can be interfered if the patient had endogenous anti-globulin antibodies, which can be found 15 to 25% of the patients, and it can be result in falsely low levels of the thyroglobulin. Second is the thyroglobulins had low sensitivity during thyroid hormone suppression therapy, so the clinician cannot differentiate by the thyroglobulin level whether the patients present with disease or in remission. And the final limitation is that the serum thyroglobulin had different methods of detection, so there are various sensitivity for each method. For example, radioimmunoassay or the chemiluminescence immunoassay. So due to this limitation, many researchers try to develop new biomarker instead of detecting the thyroglobulin, which is the protein level. And the serum thyroglobulin detection is based on the antigen-antibody interaction, so there are some antibody interference. So they develop new biomarker by detection of the RNA level, which is depend on the molecular technique, and it is more sensitive. The first report on the thyroid-specific mRNA for the detection of circulating thyroid cancer cell in peripheral blood was first reported in 1966 by Ditkoff et al. They used reverse transcription PCR to detect the thyroglobulin mRNA, and they showed that these assays has no interference by the TGAB or the thyroglobulin antibodies. And the assays has high sensitivity during thyroid hormone therapy. And the presence of the transcript indicates or helps identify the patient at risk for metastatic thyroid cancer. Here is the example of the thyroid-specific mRNA, the thyroglobulin mRNA itself, or second is the TSH receptor mRNA, which responds to the TSH and helps stimulate the production of thyroid hormone. Third is the thyroid peroxidase, an enzyme that convert iodine in the bloodstream into iodine, and addition of the iodine to the tyrosine residue of the thyroglobulin. And finally, it's the sodium iodide symphoretin gene, which help the uptake of iodine into the follicular cell. These two genes, the thyroglobulin and the TSH receptor, is the most studied for the follow-up of DTC. However, this is the review of the thyroglobulin mRNA by Werberg et al. In 2004. He demonstrated that the tgmRNA has various sensitivity in the follow-up of DTC from highly significant to no such correlation of this assay, mainly because it has various methods of detection. For example, the detection is quantitative or not. If you use reverse transcription, it's maybe just a qualitative detection. And then later on, many studies use the qPCR or quantitative real-time PCR, which is much more sensitive, but maybe it's too sensitive, so there's no difference between patients and the controls. From the American Thyroid Association Guideline 2009 state that tgmRNA may be a sensitive marker for persistent thyroid cancer. However, the extraction of RNA is still not well standardized, and its specificity still be questioned. So further studies optimizing the measurement are needed. So among the thyroid-specific mRNA, the TSH receptor mRNA seems to be the most effective for nowadays. The comprehensive study of the TSH receptor mRNAs are from Dr. Milas from Cleveland Clinic. Her study shows that the TSH receptor mRNA is very effective in the pre- and post-operative thyroid cancer management and also for long-term follow-up. So my objective of this study is to evaluate the efficacy of the TSH receptor mRNA in the follow-up of our thyroid patients. And second is to specifically look into the efficacy of the TSH receptor mRNA in the TGAB positive patients. And finally, to compare the diagnostic performance of the tumor marker, which is serum TG, and the novel biomarker, which is the TSH receptor mRNA, and the imaging study. We studied 160 DTC patients who follow up at our thyroid clinic, and 27 control subjects without the history of thyroid disease. Of these differentiated thyroid cancer, they are all had undergone thyroidectomy and at least one course of radioactive iodine ablation. The blood collection was done during the patient had thyroid hormone suppression therapy. We divided the patient into 4 groups. The first has 49 patients without evidence of disease. Second is the thyroid remnants based on there are imaging studies. And third is the biochemical persistent disease, which are the groups of patients with positive tumor marker but negative imaging studies. And the fourth group is the structural persistent disease, which had both positive for the tumor marker and the imaging study, which can be divided into the local regional disease and distant metastasis. The demographic of the,
Attention please, attention.
and here's the demographic of the patients. The mean age are about around 50 years old, and the ratio of female to male is 4:1, and the median follow-up is 4 years. Most of the patients, about 80%, are papillary thyroid carcinoma and follicular thyroid carcinoma and other types of DTC are found in decreasing order. We also subgroup the patients by the TGAB status, and 17% of the patients are TGAB positive. And you can see that all patients can be detected by the assay. So we apply the cutoff value of the 2 picogram equivalent per microgram of total RNA. After applying the cutoff at 2 picogram, it seems that the group of distant metastasis had significantly higher level than the patient without the disease. This table shows the TSH receptor mRNA levels divided by thyroglobulin antibody. And oh, it's all the time. And it seems that only the group of structural persistent disease and its subgroup of distant metastasis with the TGAB positive had a significantly higher level than the patient without disease. Almost done. It's almost done.
Attention please. We have an alarm condition, third floor center stairwell number 7. Evacuate the building immediately.
The diagnostic performance of the TSH receptor mRNA and serum thyroglobulin and the imaging study in identifying structural persistent disease. And you can see that for the TSH receptor mRNA, it has high sensitivity for the patient with the TGAB positive compared to the serum thyroglobulin. However, the accuracy of the TSH receptor mRNA is not better than the original serum thyroglobulin tumor marker. And from the overall results, the serum TG remains the most effective tumor marker in the follow-up of DTC. You can see that it gives the highest accuracy of 80%. So, conclusion. The TSH receptor mRNA seems to be highly sensitive in the TGAB positive patients only. And we found that 24 out of 27 of the TGAB positive patients were both positive for TGAB and the TSH receptor mRNA with the sensitivity of 88%, but the specificity is kind of low, it's just only 42.9%. And interesting that 2 of our patients with distant metastasis has unreliable tumor marker because the undetectable serum TG, and they had TGAB higher than upper limit. So these both tumor marker it's not beneficial for these 2 patients. However, the TSH receptor mRNA assay were positive. And this emphasized the serum TG remains the most efficient marker compared to the TSH receptor mRNA, which is much more expensive and time-consuming. And fortunately, the second generation of the TG assay made it possible to be sensitive enough for detecting basal TG. And so the TSH receptor mRNA may not be used as a primary test or the only test for the follow-up of the DTC, but it may be used as an adjunctive marker whose patients had serum TG not reliable due to the interference of the TGAB. My further study will emphasize on the cost effectiveness of the routine usage for the assays, specifically in the TGAB positive patients, and may extend to the novel markers such as circulating BRAF V600E in the diagnosis and the follow-up of DTC, which is not just a thyroid-specific but a tumor-specific marker. Thank you for your attention.